Thank you. Thank you. And good morning, first of all, to you who are here in person, and also to those who joined us virtually. I'm aware that some of our colleagues, notably from the US, have a hard time to do that after midnight. But anyway, thanks, therefore, special thanks for doing this. The topic here, if I can have the first slide. The topic here is actually dealing with novel vaccine platforms. And I thought it's important not only to look at newly emerging pandemics, but also on existing epidemics. And we have a wonderful group of speakers here. So I am sure this will be a very, very interesting morning session. Originally, we had planned a little bit longer talks and less discussion, but then everybody felt it's better to have longer time for discussion and shorter time for talks. So I hope our speakers all accept this. I told you 8 minutes, max 10 minutes, and I will have to wave hands so that you see that it's time to come to an end after 8 minutes or so. I will not go into the major topics, but I wanted to say clearly that COVID-19, as we all agree, has shown us the threat of newly emerging pandemics. And it has also shown everybody that it does not stop at any doorstep, that it arrives at every home. And therefore, this is clearly a demonstration what happens if preparedness is insufficient. But one thing I thought was clearly very, very impressive and quick to arise, and that was in medical intervention measures, notably, of course, vaccines, and that's what the topic of today is. We will look at vaccine development, what have we achieved, what can we benefit from these new vaccine platforms, and are they good enough also for the current epidemics such as HIV/AIDS, TB, and also for another emerging threat that has not reached us but is threatening us, and that is a pandemic flu, obviously. And I will not go into further details. We thought that we have the presentations. You have time for short, direct questions, and the rest then will be dedicated to the longer discussion amongst all of us. And as many questions as come up, I will try to get them all integrated. If not, I have a number of questions to start with if there is a kind of hesitancy in the beginning. So I was told also that you always have to use the microphone. As you know, this is live and virtual. Otherwise, the speakers who are not with us here in that room will not hear what you said. And I was also told to tell you, please don't touch the microphone, because then we lose time for hygiene measures, that is for disinfection. That's not a major issue, but since we want to get the most out of this, that would be kind if you could think about it once you are at the microphone. So without further saying, let's start. And the first speaker is from Berlin, Professor Leif Sander. He studied medicine in Hanover, and then he, I think, was briefly as a physician in Aachen, and then moved to the Icahn School for Icahn School of Medicine at Mount Sinai in New York. Came back then to the Charité, where he took over a professorship for infection immunology and vaccine research. And he has been quite vocal also in this pandemic, at least in Germany and also Europe. And I ask you, Leif, to start with your presentation.
Thank you very much, Stefan. Thank you very much to the organizers for inviting me and for putting together this most interesting session. So obviously we're talking about those new technologies, and as I'm the first speaker, I think it falls to me to just give us a brief background on where this actually started and where the idea originated from. I hope the slides are... So obviously, this is known to everybody in this session, but I still think it's a good way to start the session to remind ourselves and how quickly we reached this this goal of developing effective vaccines. So 314 days after the genome was sequenced, basically we had our first efficacy data, highly positive efficacy data, and as of yesterday, I think 6.84 billion doses of vaccines, different types of vaccines, have been administered worldwide. So that is obviously a huge success. We were lucky with SARS-CoV-2, if you wish, because we sort of knew that the spike protein would be a very good target for vaccines, and it was more of a question of how to induce immunity against the spike protein. And what we're going to talk about today, I think, is mostly those new platforms, vector vaccines and mRNA vaccines, which have really shown their potential during this pandemic. But of course, we also have inactivated vaccines, which have been used for a long time and which are being administered worldwide. The COVID pandemic, as horrible as it is, has also been a catalyst, I think, for those new technologies. And in the with the example of mRNA vaccines, we know that this has been a development that has been going on for decades. But lucky for us, I think this technology was basically ready for prime time just when the pandemic hit, and the pandemic and all the funding and impact that came with it has basically sped up the clinical development, and obviously you can see that how quickly we've now achieved several mRNA vaccine platforms that are highly effective against COVID. And this is just, I could show you a lot of different pieces of data of how well these vaccines work. And this is one of my favorites that's, I think, still as a preprint out there, but I could show you data from Germany as well. This is from the US. And if you just look at people that have been vaccinated with mRNA vaccines and or not, and the rate of hospitalization, and you can obviously see that once you're vaccinated with one of those vaccines, you have a very, very strong protection against hospitalization from COVID-19. What we also learned, and I think those different platforms that have been developed, the adenoviral vectors, as well as the mRNA vaccines, the ones that were first licensed, at least in Europe and in the United States, that we can combine those. And this is also not a new idea, but I think, again, has been brought to the spotlight with COVID-19. And this is a study that we were fortunate to do at Charité, but other studies have basically shown the same. And what you can see here is that if you receive 2 doses of the BioNTech Pfizer vaccine, you develop very high and potent neutralizing antibodies. Sorry, can you go 2 slides back, please? Yes. And the homologous immunization with 2 shots of chimp adenoviral vector also gives you robust immunity, but the combination of both starting immunization with an adenoviral vector and then coming back with an mRNA vaccine really boosts the level of immune responses that we can read out here, even beyond the homologous combination of 2 mRNA vaccines. And very recently, just a few days ago, actually, we've seen the first effectiveness data for this combination, and it's really reassuring that the combination of an adenoviral vector with an mRNA vaccine can further improve not only the immune readouts that we can measure, but it can even improve effectiveness against symptomatic infections. And these are studies that were published a few days ago, and I've put the link here. And those 2 studies involve the chimp adenovirus that's contained in the Oxford AstraZeneca vaccine, and we've seen very similar data, at least for the immune readouts, for the Ad26 vector that's contained in the Johnson & Johnson vaccine. So this is a concept that I think that has been explored for other diseases such as HIV, and we've seen now with SARS-CoV-2 that even combining different types of platforms can be highly effective. So just as summing up a little bit of what we've learned in terms of immune response and effectiveness with those new platforms, and I think we'll hear more about that in a second, is that all of them are highly effective against severe disease. And I think that's the most important takeaway. They're also quite effective against, or very effective against symptomatic disease, with some differences and probably a little bit less effectiveness with the vector vaccines, and that the combination of both can further improve that. And we see slight differences in the level of humoral and cellular immune responses that can be elicited with those different platforms. And if you combine them, it seems that you're getting the best of both worlds. Some questions remain as to the durability of protection. We've seen quite a bit of waning. We were also lucky to publish a study a few days ago where we see quite a bit of immune waning in the elderly, specifically people above the age of 70 seem to have a waning immunity. And it's, I think, currently an open debate whether this is actually a 2-dose or a 3-dose regimen, and whether a third dose that's been given after 6 months is actually required. And there are some questions still as for the immunologist to the modes of action. And so okay, almost done. So basically what I want to say is that we have these new platforms and they seem to be very, very good for these acute infections. What I would say is these more simple types of pathogens that have a strategy of getting in and getting out quickly. So they get in, they infect, and they spread quickly. And they don't have to spend a lot of cost in subverting your immune system, because as soon as they're in, they can basically infect the next person. And so strong immunity against individual targets such as the spike protein is probably sufficient and will give you a good vaccine. First, with those persistent infections that have more complex life cycles and high variability, basically a model is to get in and stay, and they've spent a vast part of their evolution in subverting the immune system, and so the same strategy will probably not work. So in my take, it's not all, we can't solve all those problems with a new vaccine platform, but we probably have to combine different strategies here. And last but not least, we have pathogens in which a single antigen doesn't exist. We also have pathogens where probably protein antigens, and both the vectors and the mRNAs are designed to target those protein antigens, probably won't do, and we need carbohydrates, lipids, and other targets. And so we need new platforms and new technologies also for those non-protein targets. And I'm skipping those 2 slides now for time, but there are new technologies such as synthetic carbohydrates, and we're working on this with the Max Planck Institute. And I'm skipping those slides for time reasons. But I think this is something we can discuss. And so finally, I think those new technologies will probably be the choice for future outbreaks and pandemics. But we need more beyond that, and I think that's something that we can discuss. I'm not reading this, but we need additional platforms and new strategies to combat other types of infections, more complex infections such as bacterial and fungal and parasitic infections. Thank you.
Thank you, Leif, for this interesting talk. As we said before, specific questions can be asked now very briefly, and otherwise for the general, like how can these platforms harnessed for other diseases which are probably more complex and include bacteria, fungi, or very tricky viruses such as HIV. One question that I would like just briefly ask is, the studies you showed were not specified, or were they also clearly showing whether it's Delta or another variant? Or is it most likely to be Delta? I mean, there was a study very recently from Scotland in the New England Journal looking at the Delta variant and effectiveness of the vaccine, because that's the question of with the second boost or whatever.
I think we have to we do see that Delta basically decreases effectiveness. I think that there is consensus on that. But in addition, we also have the time issue that with a longer distance to the second jab, basically, that immunity is starting to wane off, which is also a natural phenomenon. But I think in combination, this could lead to those vaccines needing a third shot for at least vulnerable populations.
Thank you. Thanks very much again. And we come back to all these general issues, hopefully with sufficient time at the end. Next speaker is Wayne Koff from the Human Vaccines Project. And he joins us virtually from the US. So again, thanks for staying up or waking up early. Or staying up long, whatever. Wayne is the CEO of the Human Vaccines Project. Before that, he was with the International AIDS Vaccine Initiative. He has a long-standing experience in understanding vaccinology from the basic immunology point of view. And the major point, I understand it like that, is that from the Human Vaccines Project, which he founded, is really to rationally design vaccines on the basis of a best possible understanding of the immune system of the human. And I won't go into further details. Wayne before was at the National Institute for Allergy and Infectious Diseases. He was in a private company before he went to go to ERV and then later to HVP. And he studied medicine at Washington University and Baylor. Now, Wayne, the stage is yours.
Thank you, Stefan. It is really wonderful to be here, albeit in the middle of the night. Well, as life has indicated, this has been a great success story in the vaccine space, where we have a number of vaccines that have been developed within a year. But even with that, as we all know, we still have the hundreds of millions of the cases and the millions of the deaths. And as we were speaking about offline, it appears that we're at the beginning of another wave. And so one of the take homes here is that we have to understand and see if there's a way to get out in front of the curve. As Life also said, this is an extremely easy virus to neutralize, and we got lucky. I think it's unreasonable to assume that for diseases like HIV and malaria and TB and some of the complex diseases that it would be as simple. And so one of the issues that we've focused on in the Human Vaccines Project is to really understand how the human immune system is working, with an aim of really getting out in front of the curve. And what I mean by that is there a way to develop the universal vaccines against the coronaviruses, and potentially against the influenza virus and some other diseases that we worry about. So the epidemic has given us a number of the opportunities here. And one of the opportunities is that it's the only time really where we have a series of vaccines that have been developed that have a similar antigen in a variety of the contexts, whether it's the mRNA or the adeno vectors, or the adjuvanted vaccines such as the Novavax vaccines, and even some of the peptide vaccines that are coming along. And of course, we also have now the early stages of development of live attenuated vaccines actually for this disease, and we have the inactivated vaccines. At the same time, we are at a unique opportunity because we have a series of tools that we have never had before in terms of the analysis of human immunity, systems biology, AI, and machine learning. And we also have a unique situation, as Life indicated. We have 6.8 billion of the doses that have been given across the world. albeit in somewhat of a non-equitable way. We still have a limited amount of access of these vaccines in Africa and some other areas of the developing world. And so what we're doing here at the Human Vaccines Project is looking at the immune system in terms of ways of understanding it, to extend the ability and the efficacy of vaccines, and particularly in those individuals that are most vulnerable, which in this case is the elderly. In other cases with other diseases is the maternal infant space, and those living in the developing world. And so this is key. We have tools such as a series of omics assays, the metabolomics and transcriptomics, lipidomics, etc. And we also have sequencing ability of the B cell repertoire and the T cell repertoire that we never had before. This has yielded us in the Human Vaccines Project and a variety of other teams the beginning of understanding some of the markers, the biomarkers that are the predictive markers of reactogenicity and immunogenicity, efficacy, and soon to be a better understanding of the durability of the vaccines. So I think this is as a take-home, if you will, has put us into a situation where we can, as a vaccine field, begin to think about the concept of more of the universal vaccines, and a number of groups are beginning to look at this. In one of the op-eds that we wrote in Science about six months ago, we highlighted that it's never is too late, in a sense that there are hundreds of the coronaviruses, if not more, that are in the animal models at the moment. And so we should have an increased emphasis on looking from a surveillance mechanism. I know that one of the other of the talks is going to be in the diagnostic space, and really understanding, which of the subsets of the viruses have the potential of jumping into people and expanding either in terms of the epidemic or as a pandemic. This is a unique opportunity. I think it's unrealistic to assume that any of the individual platforms is a panacea for everything. And certainly in the HIV space, where I spent a number of years, looking at development of HIV vaccines, it still is really an antigen discovery problem. So I think I'll stop there, and I believe at the end of the day, the combination of the platforms, as Life has indicated, is really going to yield us unprecedented opportunities in the future. Thank you.
Thank you, Wayne, for this kind of overview on where we are going in the future with the analysis of big data, big immunology data. Are there any specific questions at this stage? No, I will come back with the major question for me on general terms, more how to get predictive markers of vaccine protection with probably you will need the vaccine trials first and then go on, but we come back to that if that's okay for you. Next speaker is Nathalie Garçon. She's now the Chief Scientific Officer from BioAsta in Lyon, France. And she has indeed studied in France, made 2 PhDs, one in pharmacology and one in toxicology with immunologic aspects. Then she went to the UK and afterwards spent a couple of years at Baylor, Houston. And mostly then I know her from her time at the GSK, GlaxoSmithKline, where she was the expert of the AS the adjuvant system development, which was so successful in some of the more recent vaccines of JSK, notably the herpes zoster vaccine. She has then, as I said before, moved to form the BioAster, a scientific technology research institute on infectious diseases, microbiology. And she will talk also on platforms in the context of pandemics and highlight the chances, but also the missed opportunities. Natalie, the stage is yours.
Thank you very much, Stefan, and thanks for inviting me to present on the platform. Can I have this slide? Oh, I'm the one to share, sorry. Okay. Oh, sorry. Doesn't work.
Yes.
No, I don't have the. Okay, you see it?
We see it.
Okay. So what I would like to do to talk about, as Stefan said, is actually the new vaccine platform and somehow the chances and the missed opportunities that occurred during the COVID crisis. So if we look at the different vaccine platforms that were activated in the context of COVID, you basically span across the whole type of vaccine that has been used from the beginning. The first pack somehow are the ones that are based on the whole microbe approach, where the first one were inactivated vaccine and the first one was cholera. Now it's polio and flu to some extent. The second one is live attenuated vaccine, so they are not killed, but they are attenuated so that they do not induce the disease. And the first one was the rabies vaccine. And today, mumps, measles, and rubella are the flagship vaccine for those type of this type of approach. The third one is what we know today as live viral vector vaccines that have been being developed and tested for a long time, actually. But the first time they were licensed, and that was on a specific condition under Ebola. Two different live vector vaccine were licensed, one based on VSV and the other one on adenovirus. And then the next platform that was developed and that was for the first time licensed through an hepatitis B vaccine was in '86 with what's called the subunit approach. So we talked earlier about the antigen and the criticality of the antigen for the induction of a proper immune response. The principle of subunit vaccine is to extract from the virus the antigen which is of interest, and in the context of recombinant antigen, they can be produced as very high quantity under specific conditions. And finally, the last type of vaccine that has been developed is the one which is based on genetic material, and this is DNA or RNA vaccine. They have been in the making since more than 50 years now, and the first licensure occurred last year with mRNA vaccine in the context of COVID. So why so many vaccine platforms? Where they were developed because they were answering to the need of improvement of vaccine. The first vaccine when they were inactivated were actually by chance, like the design of those early platforms. So there was a need to improve on the immunogenicity of those vaccines. As they were clean, and not replicating, the immunogenicity could be not sufficient. However, the live attenuated vaccine had other potential issues that were in particular the reactogenicity that they induced, and the fact that one was to be developed for each. And here come the live viral vectors, where basically you have one virus that can fit all needs of vaccine, and where the antigen that is of interest is within the genome of the genetic material of that virus and can be expressed within the organism that gets the live vector. However, those have been, there are questions, and there were questions that existing on the live virus. Can you give them several times? Can you what would be the type of reactogenicity. Parallel to that, the subunit approach was developed, and the big advantage certainly of subunit vaccine is that you can mass produce vaccine because you can produce them in through fermentation, through cell culture. That doesn't present the pitfall that you may have with using the original virus. They do, however, require adjuvants since you're missing the signal that induces the innate immune response critical for the development of a persistent immune response. And finally, the mRNA vaccine, and one should not forget that originally the big and certainly the major advantage that was seen by that technology was the ability not only to mass produce, but to fast produce. Those vaccines can be produced in a record time versus what you can do when you have to do cell culture or fermentation. There are still pending questions, clearly, since they have been from mass production to mass administration in a record time. And there's still work to be done on the ability not only to increase the immune response, but the persistence of that response, and also the reactogenicity that can be seen. So if we look at the COVID vaccine that have been used, I think we can see advantage and disadvantage on each of those platforms. In terms of production, live attenuated, inactivated, or viral vector, they are known process that have been used from decades. The limitation clearly is the capacity to grow the virus when we're talking about viruses that are pathogenic. So you need to have a BSL-3 production facility. When you look at subunit approach, again, they have a known process. They have been used in decades. They do require an adjuvant. In the context of a pandemic, the obvious question is, will enough adjuvant be available? And finally, for the mRNA vaccine, clearly they are produced very fast. Raw material availability can be a question. And as we heard already, could they be universal, which clearly is wishful thinking. In terms of safety, for the whole virus microbe approach, you do have the safety that you expect for attenuated and inactivated virus. They can be reactogenic. There are serious adverse events that have been described for the context of live vector. In the context of subunit approach, in the context of COVID, they are not yet registered. What has been seen and using adjuvant is that you can have a reactogenicity depending on the adjuvant. However, could this approach be the solution for boosters? As Wayne said, equity of vaccination become critical, and not only a mismatch of approach can be of value to increase the immune response, it can also be of value to allow for more people being vaccinated. And finally, for the mRNA vaccine, there's a serious adverse event that have been described, the immunogenicity, the persistence and the breadth of the response, as well as the population specificity remains a question in all cases. So if we look at the future, clearly, the question, and that was touched on from every speaker so far, where do we go from there, and which vaccine platform or platforms for which disease, in which population? Tackling naive individual in the context of pandemic is not the same that tackling prime individual or persistent individual. So should it be possible to decide before and at the start of the next pandemic what the platform would be for which population and in which terms? Thank you.
Thank you. Hello. Yeah, thank you, Natalie, for this highly interesting overview into the different platforms and their potential value. Are there specific questions? You have to use the microphone, please. Perhaps you can state your name. Otherwise, Natalie won't understand what you want to ask. There's one question from the audience.
I'm Jean-Paul, Dr. Jean-Paul from Maltesa International. I'm from Congo. Thank you, Natalie, for the great innovation technology. But I want to say, what is the incidence for the serious adverse event now, like thrombosis and anaphylactic shock for all vaccinated today?
So you got it, the series of
So
events.
the incidence, globally, the incidence is very low. And actually, this is the curse and the benediction of the pandemic. You deploy technology platform at a speed and in an amount of individuals which is never seen with other vaccines. And those actually serious events has to be taken into consideration versus the epidemiology of those diseases. And I'm not sure we do have that for every single one of them, but the incidence is very low.
Yes. So the message was clear. There is low incidences, and in general, they cannot fully exclude it. Anyway, that's how I interpret it and think about it as well. Thank you, and thank you, Natalie. Thank you for the question. And we go on with our session with Dr. Anne Ginsberg from the Bill and Melinda Gates Foundation. Anne did her MD at Columbia, PhD at Harvard and Washington University. He was a chief medical officer at the NIH. Then she went from academic research into private research at Merck, and then came back to the Global Alliance for Tuberculosis Drugs, and afterwards came to the ERAS Foundation, which is was a foundation to develop tuberculosis vaccines, and she's now Deputy Director of Tuberculosis Vaccines in the Global Health Division of the Bill and Melinda Gates Foundation. She has a great overview of all the different types of vaccines in the area of TB, but also beyond, and she will talk actually about this issue, namely the status of TB epidemics, but most clearly also of TB vaccine development today. And thanks for joining us after midnight.
Thank you, Stefan. It's a great pleasure to be joining you at the World Health Summit, even if virtually. And I have, as Stefan said, been asked to tell you about progress in TB vaccine development, in particular with some focus on the use of viral vectors and potential use of mRNA platforms. So why interest particularly in TB? Well, TB remains one of the leading causes of death in the world today, and the leading infectious killer due to a single pathogen, with the exception, of course, of SARS-CoV-2. The WHO has just released their 2021 Global TB report and have announced that there were an estimated 1.5 million deaths due to TB in 2020. This is the first time in a decade in which the number of deaths has increased over the previous year. There were also an estimated 10 million new cases of TB amid reduced access to diagnostic, treatment, and preventive services, due largely to the pandemic. So it's clear that new tools and improved vaccines in particular are critical to—
Excuse me, and do you want to show slides? I mean, PowerPoint?
Are you not seeing my screen?
No, we don't see those. I think they will arrange in a second. Sorry for interrupting, but I know you sent me some slides. So
Yes, I was sharing, but okay, let me try again.
Yes, please. Coming.
Sorry about that.
You have to go in full screen.
Yeah, they are in full screen on my my monitor. They're in full screen. Still not seeing them?
We see slide 3 of 9, new vaccines, but not in full screen. That's
okay. I don't know why they're not in full screen.
Sharing the wrong screen.
You're sharing the wrong screen, I heard, but I...
Huh. Okay, let's try this then.
Wonderful.
That's good?
Perfect.
Okay, sorry about that. All right, as I was saying, I think it's clear that new tools and improved vaccines in particular are going to be critical to ending the TB epidemic. Now in TB, transmission is mainly from adolescents and adults, really overwhelmingly, because those are the ones with active pulmonary TB. In the last few years, it's been recognized that TB is actually a spectrum, from initial exposure and then infection and a latent infection to disease. But there's also a state of subclinical TB, seen here, highlighted now, in which individuals are asymptomatic, but MTB can still be detected in their sputum. We don't yet know how much of transmission can be attributed to this group, which is quite prevalent, in fact. Up to 50% of people with MTB in their sputum actually are completely asymptomatic, and in some studies even higher. There is only one globally licensed vaccine for TB, Bacille Calmette-Guerin, or BCG, and it's been in use for 100 years. It's delivered very widely, but primarily, almost entirely to infants in high and moderate TB burden countries. And while infant BCG protects young children under 5 from severe forms of extrapulmonary TB, it clearly does not effectively protect adolescents and adults from developing active disease and transmitting it to others. So the primary target for new TB vaccines really is this adolescent and adult age group, because we have to stop TB in them to stop transmission and to protect all age groups. So what does progress look like recently in TB vaccine development? This is the current global clinical pipeline. As we know it, there are 14 candidates in clinical development, and they take advantage of a range of platforms. There are 3 that are virally vectored, all in early phase 1 or early phase 2 developments, shown here in the dark blue boxes, and taking advantage of adenoviral vectors or flu vector in one case. And then there are a number of adjuvanted recombinant protein vaccine candidates in the bright blue, and several of those are in efficacy trials, phase 2B, and the M72 AS01 vaccine has already completed a phase 2B, and we'll talk about that just briefly in the next slide. And then there are a fairly large number of candidates that are based on mycobacterial, whether killed, whole cell, inactivated, live, attenuated, or extracts. Now this portfolio not only is somewhat skimpy, certainly compared to the COVID-19 portfolio in terms of number of candidates, but it also is very limited, certainly amongst the subunit vaccines, in the immune responses it's meant to induce, which are almost entirely Th1 CD4s, and in the MTB antigens that are expressed by these subunit vaccines. And so it's a fairly narrow portfolio and not nearly as robust as we need it to be. And we know already that while Th1 CD4 responses are likely necessary for protection in humans from MTB, it's quite clear that they're not likely to be sufficient. And so there's a lot more work that needs to be done. You can also see in these white labels that there are a number of different kinds of endpoints being explored in the efficacy trials with these TB vaccine candidates, including prevention of infection, prevention of disease, which is really the endpoint that we need to meet in order to have the greatest impact on the epidemic in the shortest amount of time, and prevention of recurrence, because there's a high rate of TB recurrence in recently cured TB patients. Now, there has been, despite the limitations of the portfolio, which I've just discussed, some really exciting recent progress in this field in the last few years. And that includes these 2 positive efficacy trials in particular. The one shown on the left, published in 2018, was a study of a novel subunit vaccine, H4IC31, which did not show statistically significant efficacy, but also a BCG revaccination compared to placebo in adolescents who are at high risk of becoming infected with MTB, but are not yet infected at the time of vaccination. And revaccinating these adolescents with BCG provided about a 45% decrease in sustained MTB infection measured with an interferon gamma release assay, which we believe equates to essentially latent TB infection, LTBI. Even more exciting, perhaps, was the study on the right, a phase 2b proof of concept trial of the novel subunit vaccine from GSK, M72-AS01E. This vaccine was evaluated in adults in Africa who were already MTB infected at the time of vaccination and provided about 50% protection against active TB disease. So that has injected really enormous energy and optimism into this field. And just to mention that as an example of how TB vaccines are definitely leveraging learnings, both platforms and adjuvants from other fields, the AS01 adjuvant that is such an important part of this novel subunit vaccine is the same adjuvant being used in the malaria vaccine that was recently recommended for use, RTSS, and in GSK's very effective shingles vaccine. So these efficacy trial results are really game-changing in a number of ways. They provide for the first time proof of concept that a subunit vaccine can protect against tuberculosis disease. And the M72 phase 2b was the first demonstration that a vaccine can protect adults or individuals of any age group that are already MTB infected and protect them from getting TB disease. The BCG revaccination, of course, provides indication that perhaps there are even better ways to use BCG than the way it's been used primarily over the last 100 years. And these studies, because biorepositories were created during these efficacy trials, they provide us really the first opportunity to discover at least candidate correlates of protection and hopefully increase our understanding of protective human immune responses. So there's also been exciting progress in the last few years in the preclinical and translational sciences space. And again, borrowing from other fields, this includes viral vectors, in particular, for example, a CMV, cytomegalovirus vector TB vaccine. And since 2019, in a project at BioNTech supported by the Gates Foundation, there's approaches to applying mRNA vaccine platforms now to TB protection as well. There are also a number of studies trying to look at alternate routes of administration for this airborne disease. It may well be that mucosal or aerosol administration will be important, and that's been looked at least one phase 1 clinical trial. There are a number of other new tools available, exploration of novel adjuvants in this field as well, and importantly, exploration of more diverse vaccine-induced immune responses. But the question I get asked most often these days is, despite these recent advances, why is vaccine research and development taking so much longer for TB than it did for COVID-19? And in my view, there are really a number of important factors to this, and probably the most important group are those related to the pathogen itself and the nature of TB compared to that for COVID-19. So TB being a very complicated and large bacterial pathogen with about 4,000 antigens compared to a much smaller, simpler virus. And then the nature of the disease, which is a much more chronic and slow to develop infection, means that clinical trials are going to take a much longer period of time until we have a correlate of protection, which is why this discovery of correlates is now a major emphasis in the field. And importantly, because of the difference in distributions between COVID-19 and TB, where TB is really largely limited to people living in poverty in low-resourced or a few middle-income countries, compared to SARS-CoV-2, which of course has spread globally, including to the wealthy nations, there's a vast difference in the sense of urgency to attack these diseases and to develop the new tools, including about 1,000-fold differential in the funding resources made available over the last year or two. So I just want to thank you for your attention. And of course, I look forward to the coming discussion. Thank you.
Thank you, Anne. Thank you for the overview. Are there burning questions? No. So we will, from the audience here and also from the virtual arena. So let's go on to the next talk. We're moving now from vaccines to diagnostics, and I guess everybody that we need the three major pillars of intervention: vaccines, diagnostics, and therapeutics. And we thought it is really worth also one talk on a non-vaccine approach, and that is diagnostics. And particularly we here in Europe and Germany know the importance of diagnostics, also routine diagnostics for the control of COVID-19. And our speaker is Dr. Bill Rodriguez from FIND, where he is now the Chief Executive Officer since a couple of months, if I understand correctly. So Dr. Rodriguez studied medicine in Yale, did his training at the Brigham Women's Hospital in Boston. He was involved in the Bill Clinton Foundation in pricing of HIV/AIDS drugs, antivirals, and as I said, he's now the CEO of FIND, and he will convince us that diagnostics need to be linked with vaccines in a successful pandemic response.
Thank you, Stefan. Good morning, everyone. For those of you who are not familiar with FIND, we're the Global Alliance for Diagnostics, and we work to ensure equitable access to testing around the world in service of the greater goal, which is better health for all. Like Gavi and like CEPI, we play a coordinating role connecting countries and communities, the funders, decision makers, healthcare providers, patients, and then especially industry, new platform developers who are working to spur new innovations that will allow testing to become more integral in sustainable and resilient health systems. We've been the co-convener of the ACT Accelerator, the ACT-A Diagnostics pillar alongside the Global Fund through COVID, so we've had a unique window on the challenges and opportunities in rolling out new tools for pandemics. Let me just start by saying that I'm quite happy to be here, and even though I'm not in Berlin with many of you, I'm grateful that the meeting's taking place in Germany, which has been a leader in global health with a long history of championing the global response to pandemic threats and pandemic preparedness, including longtime support for FIND, for which we're grateful. And I think this is reflected in the fact that Berlin has been chosen as the location for the World Health Organization's new global hub for pandemic and epidemic intelligence. And so we're looking forward to working closely with Dr. Chikwe Ekekwezu and collaborating with our partners around the world on predicting, preventing, detecting, preparing for, and then responding to pandemic and epidemic risks worldwide. Like Stefan said, I think my job on this panel and this morning is to serve as a bit of a counterpoint and a reminder that vaccines have a context in how they're delivered worldwide, and just to temper slightly the enthusiasm for really the breathtaking work we've seen. I think Leif mentioned it in the first talk. It's really remarkable what the world has seen in the past 18 months with respect to vaccines and vaccine development and the potential for the future applications of new vaccine technologies. But I want to also note the challenges that we've seen in the realities in countries worldwide and also how that relates to testing. So next slide, please. So let's just review what we've learned from COVID to date about vaccines in the reality on the ground. And the first thing to note is vaccines have been essential for the response to COVID, but they're not a panacea. And I think it's hard for all of us to remember way back to March and April of 2020, at a time when vaccines were still in development and aspirational, but there was an urgent crisis in testing, and there was no access to tests that could reliably diagnose people with COVID and SARS-CoV-2, and a lack of availability around the world. That crisis from a year and a half ago has been overtaken now by the crisis in vaccine equity and vaccine availability. But it turns out the testing crisis was never fully addressed. And so today, much of the world is still in the midst of this pandemic, and we are all nervously expecting yet another surge from Delta or potentially a new variant. And we know that many countries will not have access to these tremendously effective vaccines for several months or even years. So for instance, you've probably all seen the recent report from the WHO that the 10% target, the actually quite low, painfully low target that all countries should have 10% of their populations vaccinated by the end of September, was missed by 56 countries, and most of those in Africa. And the target of 40% vaccination coverage by the end of this year is at grave risk, mostly in those countries. But for those of you who have been focused on vaccines throughout this pandemic and prior, I'm just here to tell you that you're in good company, because vaccine targets are not the only problem. As poorly as we're doing globally as a global community on making vaccines equitably available to everyone, we're doing just as poorly on the testing side. And I'm afraid soon we'll be in the same situation with the therapeutic inequity if molnupiravir and other oral therapies prove to deliver on the promise that we've heard from the preliminary reports and preliminary data. So if you look at the bottom of this slide, we've been working with our in-country partners, with the ACT Accelerator, with the WHO and global stakeholders, and we've collectively set a minimum target for testing for the next 12 months worldwide, which is to have a testing rate of 1 test per 1,000 people per day, or 100 tests per 100,000 people per day, which is what's shown in the graph. And then linking those people who are tested to the appropriate public health interventions, whether it's access to vaccinations if they've lacked them, clinical care, oxygen therapy, early therapy with effective agents, and public health interventions like contact tracing and isolation. So these targets of 100 tests per 100,000 per day are equally unambitious to the vaccination targets that we've set. So for instance, if we hit this number in Africa, if we jump from 16 per 100,000 people per day to 100 per 100,000 people per day, we'll still be at less than 15% of the rates that have been set in Germany, across Europe, and across high-income countries worldwide. Why is that relevant for vaccines? Well, keep in mind that for every country you see on this map, there's a surveillance program in place where maybe 1 to 5% of all positive SARS-CoV-2 tests are sent for sequencing and then for variant detection. So less testing at the rates we've talked about means less sequencing, and that means less timely understanding of the emergence of new variants, and we'll be behind on our ability to respond to variants that require new vaccination, new vaccine formats. Next slide. So 2 points to make, and I'll be brief. So the number one is as with vaccines, we've seen an equally breathtaking response in the development of tests. So the rapid deployment of rapid antigen tests and of molecular-based PCR tests and of sequencing has been essential to the response. And those tests were available for rapid tests within 60 days of the declaration of a public health emergency, and for PCR within 60 days, and for rapid tests within 200 days. So that means that high quality, accurate tests, which have supported vaccine studies, efficacy studies, the vaccine rollout, and have been critical to allowing sequencing to detect and monitor the emergence of variants, really came much more quickly than we've ever seen in the diagnostics industry, similar to what we've seen in terms of the pace at which vaccines have become available. So the issue is that we need effective, connected surveillance systems to continue to be coordinated at a global level in order to link the testing response with the vaccination programs, not just for SARS-CoV-2, but for other endemic diseases, and also for new pandemics that we expect could emerge in the coming years. So we're hopeful that Berlin and the new hub will play a critical role in coordinating this effort, because that surveillance is so essential for us to understand the task at hand. Next slide. So how do we link these incredible developments in vaccine development, in test development, and the challenges in driving equity and making those available for SARS-CoV-2, but also for the future? If I had more time, I could talk about some of the work we've done to link diagnostic responses to endemic diseases like yellow fever, where testing and surveillance have been essential to understand where to roll out the existing vaccines and for vaccine implementation strategies. But let me just focus on pandemics. And it's a real pleasure for me to be sharing this stage today with CEPI and my colleague Mel Seville from CEPI, who'll be speaking next. CEPI and FIND have been closely involved in the development of a 100-day mission, which Mel will describe in more detail, which has been put forward with the G7, which links vaccines and therapeutics and diagnostics development to ensure that the next time we face a pandemic, we're in a position to develop and deploy the tools even more quickly and before a virus can take hold the way SARS-CoV-2 has. Coordination among vaccine developers of any platform and test development and deployment will significantly enhance that response in the future. And that also includes ensuring ongoing support and coordination for the R&D that has to happen in between pandemics, so that vaccine developers and test developers and antiviral and antibacterial therapeutics are all working across the same set of diseases with the same linked goals in mind. And one final comment before I pass off to Mel, which is one of the critical issues we've seen that still needs to be addressed is boosting capacity for fostering local innovation and regional innovation in the countries where these diseases are likely to emerge, and for production of these technologies. So we've heard a lot about vaccine manufacturing being limited to the global north. Similarly, diagnostic tests are predominantly manufactured in the global north, and we need those manufacturing sites to be equitably distributed worldwide across Africa and across low and middle income countries in general. So FIND and CEPI are working together to map out the diagnostics component of the 100 days mission that would allow vaccines, therapeutics, and diagnostics to be developed even more rapidly and to be aligned in the approach. And so with that, let me actually pass the baton, Stefan, over to CEPI and to Mel to speak about that 100-day mission and what it means on the vaccine side. Thank you.
Thank you. Thank you very much, Bill, for this overview and stressing the importance of other intervention measures, and that they all have to work together like pieces in a puzzle, more or less, and not separately. I think that's the important message that I personally at least see. Bernie, I'm not sure whether it's me, but I have my distance. Okay, thank you. So no direct specific questions. So we come to our last speaker, and that's Melanie Serville from FIND. Sorry, I was too much in FIND, and that's why I kind of. But you're close working together. It just illustrates how important interactions are. Anyway, Melanie Serville from CEPI, apologies again. You've studied medicine in London, UK, and then had kind of a mix of academic, but also public-private activities. I understand you were with Janssen on all these difficult beasts, including Ebola, dengue, mostly viruses. And finally, we now see you here at CEPI as the head of clinical development first, and now director of vaccine research and development. So you are really ideal candidate for discussing how we can build on the experience from COVID-19 and to respond more effectively for future pandemics. And I would like to add also current epidemics, but yeah.
Okay, great.
Thank you.
Thank you very much. If I can have the slides and actually the clicker. Thank you. Thank you, and it's a pleasure to be here, and coming last can be quite an issue because so much has been said, but maybe an opportunity to bring some of these very important threads together. So just for those of you who don't know about CEPI, so CEPI, the Coalition for Epidemic Preparedness Innovations, was formed back in 2017 following the Ebola outbreak in West Africa in 2014-2016, with the goal of accelerating vaccine development for emerging infectious diseases. As a consequence, we're working on a number of priority pathogens, had 5 priority pathogens: MERS, Lassa, Nipah, chikungunya, Rift Valley fever, where we partner with a number of developers working on a number of different platforms in advancing vaccine development, as well as working on rapid response platforms. So as an organization that is to respond to outbreaks, when we saw the emergence of this new coronavirus back at the beginning of last year, clearly we need to respond with our partners in developing vaccines specifically. And in that regard, really just to highlight some of the key aspects which allowed for the rapid development of vaccines. One critical factor is obviously the other coronaviruses. And it was actually Barney Graham's group in NIH at the Vaccine Research Center that really spearheaded work on antigen that was really critical for SARS-CoV-2 moving forward, really understanding the importance of the antigen in the pre-fusogenic state and working on MERS in one of those, as well as CEPI funding a number of developers on MERS vaccines, really gave everybody a head start in terms of understanding the coronaviruses. The second critical piece, obviously, is developing rapid response platforms, which again is another priority that CEPI had. So looking at technologies such as mRNA. So really bringing that together. And as I said, CEPI as an organization for response responded very rapidly with a number of partnerships, building on antigen, building on platforms within 15 days of the sequence being identified. We actually put this slide together right at the beginning of the pandemic as we wanted to grow the portfolio of vaccines that CEPI was funding and partnering with. We now have 11 vaccines across a broad range of platforms, either in development or actually being rolled out for use. Natalie's pointed out some of these points previously, really looking at the key attributes here of speed, cost, scalability, and usability for each of the platforms. And I think this is pretty much born to what we have seen. We have seen some very rapid response in terms of viral vectors. And University of Oxford always had in mind using the ChAdOx technology for rapid response. And obviously as a viral vector, were able, as was Janssen, to move their candidates forward very rapidly. In terms of speed, definitely RNA was there. And before the pandemic, really an unvalidated platform for infectious diseases. And certainly with Pfizer-BioNTech and with Moderna, we've seen the validation of that platform. I think it's fair to say that you cannot do everything quickly, and obviously not having the time to really think about the thermal stability and the implications of rollout, especially in some lower and middle income countries. That is something that needs some significant work to make that viable in a lot of lower and middle income countries, together with the need to reduce cost. We have DNA vaccines, and we have one emergency use licensure of a DNA vaccine in India. The recombinant proteins, we scored these very low for speed, and what we have seen, we have invested because of the large scale, and we've seen two vaccines, Novavax and Clover, with excellent efficacy data, but still some work to actually get those through into emergency use and large scale use. But with their 2 to 8 degrees and strong efficacy results, likely to have an impact once they can get over the regulatory hurdle. And we've also seen inactivated vaccines move forward very rapidly. So then a little bit more on forward thinking. We know the pandemic isn't over, but we need to already be thinking about the next pandemic and moving forward. And building on what Bill said, we do have the 100-day mission. And this slide is really just to illustrate the what if we had managed to do this in SARS-CoV-2. On the top here, you have the WHO figures for cases, for fatalities, and you can see the extraordinary work with BioNTech. I think it was 314 days to submission for the regulatory, and by the 8th of December 2020, the first vaccines were being given. But there are over 67 million cases at that point. What if you could do that in 100 days from the sequence? We would have been in a position where there were only 2.3 million cases of vaccines. So how can we get close to this? And one of the first things that CEPI is doing is looking at cataloging the innovations that actually occurred in COVID-19, there were a huge number of innovations. And we have to remember that mRNA vaccines were not validated. So the speed they move forward with, there is, I'm sure, opportunity to potentially move forward much more rapidly. As well as those innovations, we need to think about what can we do up front before the pandemic hits on the platforms. Can you build platform data to move forward much more rapidly, safely, and effectively in a pandemic situation? Now, obviously, moving forward with using a vaccine within 100 days, you cannot wait to understand whether there's a real problem or not. Within 100 days, 2.3 million cases. If we'd waited until we'd known what we were doing, we wouldn't have got there. So it does require working at risk to really make vaccines the most effective in these types of situations. So from a vaccine perspective, there are 4 sort of interrelated strategies, and as we call it now, to really look at the disease X. So the first piece is really important to have the early warning systems and good, robust early warning systems to really identify what the potential epidemic or pandemic threats may be. So this really requires the vaccine experts to be working with those in early warning systems to ensure that they continue to be developed and are robust systems so that they can alert developers when needed. The second piece, and again, this was coined initially by NIH, but really looking at vaccine families. What are the most likely families to have a threat? And to build vaccine libraries of those families on the rapid response platforms so that we understand what will be the best approach to any particular vaccine in a family if it emerges as an epidemic or a pandemic. And I'll speak a little bit more about that in the next slide, but and how it can link with other work. I've mentioned the 100 days. We have a lot of work to do to look at how can we compress timelines or how can we do things differently. And then the notion of prototype targets. We've seen the success of mRNA for COVID-19. To have that as a sustainable vaccine platform, we need to take those to other targets. So known vaccine preventable diseases, develop vaccines on those and take the opportunity to further develop those platforms in terms of cost, thermostability, et cetera, and build manufacturing networks. So this next slide just highlights a little bit more the virus family vaccine, the virus family vaccine library perspective. So there are 25 virus families that impact humans. And of those, obviously, hundreds or thousands of related viruses, which may be in the humans or in animal reservoirs. So the notion of developing a library is to really sample viruses across the platform, close relatives, distant relatives, and develop vaccine candidates. So a lot of emphasis there on antigen design and putting them on rapid response platforms, taking them through preclinical and early clinical testing. So what will come out of that is not just an understanding of what vaccine will work, but also we need reagents to do immunological assays to test those potential vaccine candidates that can also be used for building diagnostics libraries. A second piece is whenever you put a vaccine in humans, you can collect the B cells and use those to develop monoclonal antibodies. So again, partnering with those with an interest in monoclonal antibody libraries so that they can use those available reagents to develop other approaches other than vaccines. So getting into the situation, so what will this actually do in the disease X situation? If you have a match, you might have a very close match to something in your library, in which case you can move very quickly, at least with a first generation vaccine from the work you've already done. You might have a closely related vaccine, which could either be monovalent or a multivalent approach in your library. Again, could be a first generation vaccine. But if there isn't a match, if you have taken the time to understand how that virus family works, what is the best vaccine approach? It may be mRNA, it may be another candidate. You can then use that, much as what happened with MERS and mRNA in COVID-19, to really accelerate vaccine development. So on the last slide here, so that was really highlighting a few of the key elements to what CEPI is looking to fund in the next 5 years. It's around our 3 main areas, which is prepare, transform, and connect. So I've highlighted some of these here, just to highlight also mentioned by Wayne and mentioned by Natalie, the potential for a universal coronavirus or a broadly protective beta coronavirus vaccine. That is also a critical area of research, with coronaviruses obviously as a huge pandemic threat. And as CEPI, we also have the need to continue to develop vaccines against our priority pathogens and bring vaccines to license. I think it's the last thing to say is we know everything is interconnected. So the importance of collaboration, not only across various researchers in vaccines, but with diagnostics and therapeutics, is also critical. Thank you.
Thank you. Thank you so much for this great overview. So this is the beginning of the kind of more general discussion. I suggest that we would focus mostly on the more scientific technical aspects. Politics comes on other sessions, for example, tomorrow afternoon on vaccine policies. So let me perhaps pick up what many of you more or less said. I start with a little bit of a provocative statement, well, perhaps, and that is, to me at least, these acute viral infections, we know for long from the immune response, there are dominant antigens. You have to attack these dominant antigens. Oops, I don't know what that is, but these dominant antigens. And there, the first question arises, obviously, there are variants. And this will be a kind of moving, perhaps then from corona, this incredible threat, to the threat of pandemic flu, where there is much more changes because of drift and shift, as you well know. So that is more dramatic. Which platforms would be good for this and that? And then move on also to the more complex pathogens, like mentioned bacteria, parasites, where there is, in my opinion, nothing like the dominant antigen, which covers, let's say, 70% of the T cell and B cell response at the height of immunity. So perhaps I start with you on the question, do you see, which platforms do you see most appropriate for the variant issue? And we know Valneva claims that their inactivated whole cell covers everything more or less. I mean, I simplify that a little bit, and then move on to the other infections. First, the example of pandemic flu threat, and then HIV, the guy that tricks out immunity in several ways, and so on, and come to other. For you, only starting the COVID issue and variants.
Okay, yes, I mean, that's actually a huge topic, and I think there are a number of different approaches that one should take. We did have already heard of the importance of the heterologous prime boost, and that is something that is likely to give you much more breadth in terms of the immune response that we are seeing from the work that's been done. So I think that is certainly an area where we need to continue to gather data to see the value of that heterologous prime boost approach. Other approaches obviously could be like flu and start looking at, do you need to develop a variant vaccine? That has actually been quite challenging because it's not really with COVID happening in the same way as flu, where one, maybe Delta now, but where one variant predominates. So I think we need to keep a close eye on that. That is clearly an option. But looking at more broadly protective approaches, either through chimeric antigens, et cetera. But we are seeing that multiple platforms do offer good protection. I think we're quite lucky with the spike protein and the ability to protect against that specifically.
So you would argue, if I understand it, you would change the kind of conformation or the sequence in the RNA to adopt for new variants, but that would not be something fits all.
So, I mean, I think there are a number of
That's
different, there are a number of different approaches. I think the antigen and how you approach the antigen, I think, is critical. Heterologous prime boost, I think, is really an interesting approach as well.
Yeah. So if we move perhaps Wayne Koff from outside from the virtual space, so would that, how would you go on then with, let's say, a pandemic flu vaccine where you have major changes and much more than in the variants that evolve through mutation only, and then perhaps say a few words whether you think there are the most promising platforms for HIV and flu, pandemic flu. Wayne Koff?
Seem to unmute. Okay, is that better? Can you hear me?
Yes.
Yeah, so I you know, it's a really interesting issue, Stefan. I mean, I think we have a number of issues here in the influenza space. We don't really have an understanding of the immunologic imprint in such that, you know, one of the important issues on flu is that it appears that the initial infection with flu as an infant, it puts you on a path towards either a better response or worse response. I think also another issue that we don't understand as well is the immunodominance of some antigens over some others. And lastly, as COVID has pointed out, here we have a naive virus that infects a range of people in different ways. And so we have the whole spectrum from the asymptomatics to, in the elderly, for example, the high levels of morbidity and mortality. So I think, as Melanie pointed out, I think we got extremely lucky here on this virus, in the sense that it was extremely easy to neutralize. We still don't understand, and the TB is a perfect example. We don't understand a lot of the correlates of the protection. As I mentioned earlier, I think we are at a place now that in the last decade there has been a convergence, if you will, of the technological advances in the monitoring of immunity, but also in systems biology, AI, and machine learning. I think the recent advances with the AlphaFold program is one example, when one can look at the sequence now and get a picture of what the antigen is looking like, that we never have been able to do before. I think opens up a range of opportunities, and as CEPI has put out a challenge, if you will, to move from a year to 100 days. The Human Vaccines Project has put out a challenge, a longer term one, which is the development of the AI model of the human immune system, and we can think ahead to a time, and we're beginning to see in the discovery of antivirals, in silico discovery accelerating in ways that we haven't ever seen. And lastly, that you mentioned HIV. HIV, of course, is the ultimate challenge in terms of vaccine development. And I think the major issue there is we don't really understand the way to induce neutralizing antibody. So we still have a huge antigen discovery issue there.
Perhaps, Anne, you favored, as I interpreted it, the M72 as an example with few antigens, and I come back to my question whether is there something like a unknown not a super antigen, because that's covered in immunology, superb antigen or group of antigens, and how do you see that? Or you think we have to go with a much broader plethora? I mean, the adjuvants, I think, have made an enormous step with the AS101. The question comes to me to the antigens here in a bacterial pathogen.
Yeah, I agree, Stefan. I think it's a crucial question that we haven't answered yet. I think it was really gave us a lot of hope when we saw that we could get 50% protection with just 2 antigens, as long as they're with a good adjuvant. Whether having a few more antigens would be enough to change that efficacy to a much higher level that we're all aiming for, or whether there are other adjuvants that need to be added, I don't think we know. I think one of the advantages of trying to apply the mRNA platform is that we can cycle through more antigens and more combinations of antigens much faster, and even try to include various kinds of adjuvants, including molecular adjuvants that may bring other features to the table. So lots
you think it's a trial, we have to go on with the kind of quotation mark trial and error, and as rational as possible, obviously. Natalie, do you think that the kind of adjuvant nanoparticle, I think nanoparticles, adjuvants have a lot of similarities, is that on the kind of height that we don't need to improve much on it? That's always difficult to say. I mean, there's always some improvement, but in my opinion, there is a big breakthrough with the adjuvants and the nanoparticles over the last couple of years.
Yeah, so actually interestingly, the nanoparticles, or the LNPs, they are used for the COVID vaccine. They originated from gene therapy. They have been designed specifically to a certain purpose, which is not actually to be given intramuscularly or target the draining lymph nodes. Yet they do function. Can they be improved? Certainly. In many different ways. The stability is not perfect. The reactogenicity or the constituent may be the source of hypersensitivity, and moreover, they do not target the cells that they should target specifically, which is the antigen-presenting cells. It's more chance and error. They are delivered and they go in the cell that they interact with. So yes, there's certainly a vast scope of improvement on those particles.
Yeah, thank you. And perhaps Leif, to you once more, coming to the complex organisms. You mentioned glycolipids also and carbohydrates. In carbohydrates, I would say the pneumococcal at least is also kind of a dominant antigen, but there are others like fungi that are more complex.
I mean, yes, I just put this out there because obviously mRNA and adenoviral vectors have had this huge success where we need to keep in mind that there are pathogens that we might not be able to address sufficiently with a protein antigen. And I think there's also been tremendous success with glycoconjugates recently for salmonella and meningococcus and so on and so forth. And I think technological development there may be moving into synthetic compounds that we can link that have a higher stability, that we can increase multivalency, would all be avenues that I think need to be kept in mind, especially for bacterial and potentially also fungal infections.
Well, thank you. Now to my last question. We're running short now of time. I have looked around, have not seen any waving hands. So, Bill, where in the research development pathway is the role of diagnostics in your opinion? We're talking about the early stage of the pipeline.
Yeah, thanks. I'll just say, there's been a lot of promise and hype around point of care testing for molecular tests and diseases, and there's been a lot of 2 steps forward, 3 steps back in some ways. But 2 things, we're at the cusp, I think, of a second wave of revolution in diagnostics. So we're about to see, in part stimulated by COVID and the research funding that's gone into COVID, the release of multiple platforms that can do multiplex detection of pathogens at the point of care for low cost and from fingerstick volumes of blood. When you think about vaccine development, I think the next step on the research side of diagnostics, as opposed to the clinical and public health side, is tests that can measure from small volumes of blood in low-cost formats, CD1 restricted responses, CD8 restricted responses, CD4 restricted responses, B-cell responses, so that the depth of the immune response to a vaccine, which can only currently be evaluated in small numbers in narrow studies in phase 1 or preclinical development, when you roll these out in these heterogeneous populations worldwide, you're now going to be able to collect that data from tens of thousands of vaccine enrollees, study enrollees. And I think that'll feed the development pipeline for vaccines quite significantly in ways that we haven't been able to do. We're still maybe 5 years from that, but I think that's the next step in collecting that data from people who are vaccinated to measure those much more granular level of immune response.
Yeah, so this would be kind of the single cell T cell assays which is a long way, clearly, but then Wayne could say a lot, but we have to come to an end. Sorry for that, but I would have liked to go on, actually. There are many, many burning questions. But anyway, thanks to all the speakers. Thank you for giving these wonderful, impressive presentations. Thanks to the audience for your interest, and thanks to the World Health Summit, and see you next time.